genetic information onto its offspring, an organism must make a copy of its DNA. The process of copying DNA is called DNA replication. During replication, each strand of the parental DNA serves as a template in the creation of new DNA. Because each newly synthesized DNA molecule is made up of one parental strand and one new strand, DNA replication is described as semi-conservative. One strand in each molecule is conserved, while the other strand is newly replicated.
Okay, so that's the basics of how replication goes. And again, this is a good slide to focus on. It tells you all the parts that play in replication and what's needed to initiate replication. Okay, so we talked about also the other functionalities of DNA besides just being replicated. So one of them is taking genes that are needed to produce proteins. And there's that two-step process where DNA becomes RNA in the process of transcription. And so we'll start there. So DNA is transcribed to make RNA. Okay, transcription begins when RNA polymerase. So what we did when we talked about replication, it was DNA polymerase that made DNA and made more DNA. So RNA polymerase starts with DNA and makes RNA. So transcription begins when it binds to a promoter sequence in the DNA. Transcription also proceeds in the 5' to 3' direction, and it stops when it reaches a terminator sequence. So you want to know promoter sequence and terminator sequence. And I'll show you how that works. So it's very similar to replication. The DNA unwinds, but again, instead of DNA polymerase coming in and adding bases at this point, it's RNA polymerase. And basically it just reads the DNA bases and starts to add RNA bases that are complementary to that DNA. Okay, so the only difference between RNA and DNA is that RNA doesn't have a T base. It has a U base instead of T. It has uracil instead of thymine. Other than that, the other three bases are the same.
Cells use DNA to store information, while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules.
Okay, so this is a DNA, purple's the DNA strand that's been unwound, and then this is RNA polymerase coming in, and this red line is the RNA, the messenger RNA that's being built off that DNA template.
Which can be used to synthesize specific proteins. Cells use three types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes.
Okay, so we're mostly going to be focusing just on messenger RNA. So basically, again, if this were a gene, this part of the DNA were a gene, RNA polymerase would come in and copy the DNA to make a messenger RNA out of that gene, and then that RNA would get processed into a protein. Okay, let's skip over that. Okay, so now we found a way to turn the DNA into RNA. It's with RNA polymerase. Now we'll talk about how does RNA get turned into protein. Okay, and that's called translation. So again, transcription is turning DNA to RNA, and translation is turning that RNA into a protein. Okay, so it's a two-step process of going from a gene into a protein product. Okay, so messenger RNA, which again is made from the DNA, is translated in codons. Okay, so we'll talk about codons. Codons basically means 3 nucleotides. So a group of 3 nucleotides, so A, C, G, whatever it may be, those 3 nucleotides together are equivalent to an amino acid that gets converted into a protein. I'll show you that. Okay, translation begins at a start codon. So the start codon that starts translation is always AUG. And it always ends at a nonsense codon. And there's 3 possible nonsense codons: UAA, UAG, and UGA. And again, we'll talk more about this. Okay, so there's 64 codons in mRNA, so if you think about the 4 different bases, there's basically 64 combinations that you could come up with of the different orders of the 4 bases. And those 64 sense codons make the 20, so there's 20 amino acids that are required to make any protein. So those 64 codons encode the 20 amino acids. So what you can see is there's more codons than there are amino acids, which means the genetic code is degenerate, which means there's more than one codon for any given amino acid, because there's 64 codons and only 20 amino acids. So there could be 2 or 3 or 4 different codons that all code for the same amino acid. Okay, and the tRNA carries the complementary anticodon, and I'll show, we have some good pictures to show what that means. Okay, so I'll show the overview first, and then we'll go into it in a little bit more detail.
Translation is the process by which the information contained in a molecule of messenger RNA is used to synthesize a protein. The process is called translation because ribosomes read along a messenger RNA molecule, translating the genetic code into the amino acid sequence of the protein. Because there are no nuclei in bacteria, translation can actually begin before transcription is completed.
So basically,
Translation is in before.
So here's your mRNA. So this is the RNA that was converted from your DNA by RNA polymerase. And then once it's formed, these ribosomes can read the RNA and make proteins, which is what is represented by these little blue dots. Each dot is a single amino acid. Proteins are made of amino acids. Okay. So it's basically reading the mRNA in sets of 3 codons, 3 bases at a time, and each base is telling the ribosome which amino acid to put there. And that determines the order of the amino acids, and then the order of the amino acids determines the functionality of the protein. So here's the genetic code. I don't expect you guys to remember this. If you're ever asked a question about it on a quiz or exam, it will be provided for you. Okay, so you don't have to know what they all mean. But basically, this tells you for all the 64 possible codons, and again, there's 4 bases, and if you just do the math, that means there's 60. You take 3 at a time, there's 64 possible combinations. So every combination is represented on this table. It tells you what amino acid that codon will lead to in a protein. Okay. So for instance, CUC gives you a leucine amino acid, CCG a proline, CAU a histidine. So these are all basically, this is how the ribosome reads the code to determine what the amino acids are in the protein. You can see at the start codon, so every mRNA has to start with AUG. Without that start codon, it won't be processed into a protein. And then there's 3 potential stop codons. So when the ribosome reaches one of these stop codons, it stops making protein. So this is the, kind of looking at it more in depth. Okay, so here's your mRNA. Okay, so again, you had a gene in the DNA. It got converted to mRNA in transcription, and then this mRNA is going to get processed into a protein. So you have a ribosome, you have these 2 subunits, a big one and a small one, and they basically sandwich on top of the mRNA. And where it starts to sandwich is based on this AUG. Okay, so it finds the AUG, and that's where it starts and comes together to start processing into a protein. Okay. Now you have these anticodons, which are these small molecules that are floating around the cell that have the different amino acids attached to them. On one end, they have the amino acid. On the other hand, they have the anticodon, which is the exact opposite of the codon. So if you see AUG, the anticodon is UAC, because that's what would bind directly to AUG. Does everybody see that? Based on, again, the rules of which bases bind to which bases. Okay? So AUG can only bind UAC. That's the only thing it can bind. So there's a bunch of these floating around. The one that has methionine happens to be UAC, so it will bind to this first position. That's why every protein starts with methionine, okay? Because the start codon is always AUG, and AUG always gives you a methionine. Okay, so it binds into this position here. It's called the P site. Okay. And then that exposes the next site, which in this case could be anything, but in this case is UUA. Okay. And UUA can only bind to AAU, and AAU is assigned to leucine based on that, again, the genetic codon table. Okay. So leucine will now fall into the second site. It's called the A site. Okay. And when it comes, the methionine and the leucine are in close combination with each other, and they basically bond to each other. Okay. So now these are your first two amino acids of the protein: methionine, leucine. After they bond, the first one basically enters the E site, which is the exit site, so it can leave, and the ribosome continues to move down the mRNA. And it exposes the third site, which is GGU. GGU binds to CCA, and CCA is assigned a glycine. Okay? So now there's your third amino acid. Now you have methionine, leucine, and glycine, and this continues one amino acid at a time until you get your full-length protein, which could be thousands of amino acids at some point. Okay? And then as it goes down, one TNA gets released and the new one comes in. Okay, so this process is just happening one amino acid at a time. Okay, it continues to grow. Continues to grow until it reaches a stop codon. Again, remember there's 3 possible stop codons. Once it reaches the stop codon, this tells the ribosome to release the mRNA, and which in turn releases the protein product at that point. And here's your new protein. Okay? So however many codons there are between the start codon and the end codon, that tells you how many amino acids your protein is going to be. Okay? In this case, it was a very small per 10 protein. That's like 10 amino acids. But again, a lot of times it's hundreds or thousands of amino acids. Okay? I know it's a lot of stuff going on. Does anybody have any questions before we move on? Yeah.
Okay, so would the promoter sequence be AUG?
The promoter sequence is not AUG, so if you look, let me go back. So the AUG is actually called the start codon. There's a little bit of material up here, actually before the AUG. So the mRNA doesn't just start with AUG. There's some stuff, this is called upstream, basically before the AUG. And the promoter would be in this region, and we'll go over that next. Okay, but this is important. There is important material here that is needed for the regulation of the gene, but it's not actually part of the mRNA that gets encoded into protein.
So the AUG is just called the start codon.
The start codon, that's where the protein processing actually starts. But again, there is some stuff before that that is important. The promoter, the repressor, there's some stuff upstream that we'll talk about. But in terms of where the protein starts, it starts at the start codon. Okay. Anything else? All right, so before we go on, let's just take a 5-minute break, and then we'll go on to regulation. So that was kind of the backbone, the mechanism of how DNA gets converted into a protein, and now we'll talk about kind of the regulation, how the cell determines when to make a protein. It doesn't just make all the proteins all the time. It has, a way to turn off proteins and turn them on, and that's what regulation is. So take a 5-minute break, and we'll come back and talk about that.